talk. It's Friday the 6th of September. Now, I've heard about this phenomena years ago, of course, but only just got some evidence to support it, so we can only just report on it now. This is from peer-reviewed literature, and it's scientists based in Japan and South Korea. And what they've done is they've taken COVID vaccines, mostly Pfizer and Moderna, and cultured them, incubated them, to try and duplicate the conditions in the human body, and they've found nanostructures have developed. Now, I don't expect you're going to watch this video or be allowed to watch this video, but I'm going to do it anyway. And if by some chance a few of you actually get to see it, then that's brilliant. Now, here's the equipment that was actually used here. It's stereo microscopes. Now, basically all this means is you're looking at it with two eyes, therefore you get stereoscopic vision. And what happened was that initially they developed two-dimensional nanostructures, and then some became three-dimensional as well. And of course, you can see that with a stereoscopic microscope. Let's go and look at some now. That was the equipment that they were using. And of course, these days, you don't so much look down the microscope, it all goes on a screen, so you can take copies of it. Now, these are from the publication. These are some of the nanostructures that were observed in as I've said before, conditions that were designed to duplicate human cells in the human body that developed from the COVID vaccines, the mRNA vaccines. Now, the scale here, we'll look, well, I'll just show you a couple of pictures, then we'll look at the scale. So these are the sort of structures that they were finding. I mean, what the heck is that? You know, that is a structure that spontaneously sort of put itself together, a spontaneous assembly of this structure from the COVID vaccine cultures. Now, the scale here, 10 micrometers. So that's so one micrometer would be the size of a sort of a bacterial cell. 7 micrometers would be the size of a red blood cell. So you can see these are nanostructures, but this is a very detailed looking structure that has spontaneously assembled itself here. Really quite yeah, well, look at it. You know, that spontaneously assembled itself. What the heck is it? Now, of course, as always, we don't have, I won't be giving full answers to these questions. Read the paper for yourself. But this means the presence of these nanostructures needs to be explained by the manufacturers and by international authorizing agencies and national authorizing agencies round the world. This is a peer-reviewed publication, and I believe it gives questions to be answered. Even if it's only that this is a load of rubbish, then that still needs to be answered. Let's look at a couple more pictures before we look at the text. So these spiral ones seem to come up again spontaneously, just put themselves together. Spontaneous sort of another spiral there, another one there. Tell you what, I don't like the idea of these spontaneously forming in the cells of my body, if that is indeed the case. We don't know that, but if that's the case, I don't like the idea of it at all. Not at all. And 10 micrometers, that's actually pretty big, actually. If that's the scale there, 10 micrometers. So this whole thing is actually quite large, relatively speaking. And what is that? Spontaneously formed structure. Well, that one. Anyway, lots more examples in the paper. Do look at it for yourself and check it out. The paper is there and the pictures are all there. Now, as I say, I don't think many people are going to be able to watch this video because I'm not optimistic about it getting a wide distribution, shall we say. But never mind, we're going to do it anyway. So real-time self-assembly, these things are bolting themselves together, as it were, of stereo microscopically visible, so you can see them through the stereo microscope. Specimens of mRNA products, mainly from Pfizer and Moderna, a comprehensive longitudinal study. So constructions in incubated specimens of mRNA products is what these workers did. And our observations suggest the presence of some kind of nanotechnology in the COVID injectables. Now, I know this sounds pretty interesting material, and I'm being very, very, very careful not to go outside of what the article is saying, the peer-reviewed article is saying. And of course, I'll be giving you full references and everything for that. So I'm being very careful in this video not to go outside what it's saying. Observable real-time injuries at cellular level in the recipients of the safe and effective Covid-19 injectables are documented here for the first time, hence the fact that we're doing it. I think this paper just came out a few days ago, last week maybe, with the presentation of a comprehensive description and analysis of observed phenomena that need to be explained. The global administration of these often mandated products from late 2020 triggered a plethora of independent research studies. Why weren't they nationally sponsored and industry-sponsored research studies? Why is it down to independent scientists? And I have talked to several other independent scientists who've come up with similar findings to this, but I can't talk about those because it's not published in the peer-reviewed literature. We're going to stick to what is in the peer-reviewed literature. Of modified RNA injectable gene therapies. So, of course, they're injecting genetic material, instructing the body to make foreign proteins. They are not injecting the protein directly. Most noticeably, those manufactured by Pfizer and Moderna in this paper. Analyses reported here consists of precise laboratory bench science aiming to understand why serious, debilitating, prolonged injuries and many other side of this particular adverse reaction occurred increasingly without any measurable protective effect. The contents of the Covid-19 injectables were examined under a stereoscopic microscope up to 400 times magnification. Carefully preserved specimens were cultured in a range of distinct media to observe immediate and long-term cause and effect relationships between the injectables and living cells under carefully controlled conditions. In other words, I'm not saying these scientists succeeded in doing that, but what they were trying to do is duplicate living conditions in the laboratory bench where they could look at it under these microscopes. They were attempting to duplicate the physiological conditions of the human body. The degree to which they did that, I'm not really able to adjudicate on, but they did their best to do that. And again, this needs to be duplicated around the world and it needs to be explained because, I mean, heck, I mean, let's look at that. I mean, look at it. I mean, what is that? What are these things? I mean, it's just, yeah. An explanation is clearly required here. I would have thought. Let me know what you think. Anyway, let's carry on. Carefully controlled conditions. From such research, reasonable inferences can be drawn about observed injuries. So they're saying that it's reasonable, the researchers here are saying it's reasonable to make inferences from what they're seeing in the laboratory to what will be happening in the body because they're trying to duplicate the conditions in the body. Of course, the studies should be done in the body as well. This should be a great trigger for future research done by authorised institutions around the world and regulatory bodies around the world. This should be done to explain this. Don't hold your breath. Right. From such research, reasonable inferences can be drawn about observed injuries worldwide that have occurred since the injectables were pressed upon billions of individuals. So they're saying that this, basically what they're saying is, I think this is a pathophysiological mechanism that could explain the adverse reactions, or some of the adverse reactions that we are tragically seeing. And of course, we've interviewed quite a few people now on this channel who've suffered from this as well. In addition to cellular toxicity, as if that wasn't bad enough, our findings reveal numerous on the order of 3 to 4. So that's 10 to the 6. That's 3 to 4 million of these artifacts per mil of the injectable. Heck, that's 4. That's 4. 1, 2, 3. 1, 2, 3. Up to 4 million, between 3 and 4 million. Dear me. Visible artifacts, self-assembling entities, ranging from about 1 micrometer to about 100 micrometers, 100 microns or greater, of many different shapes. As we said, one micrometer is about the size of a bacterial cell. If you've got good young eyes, you could probably just about see an object of 100 micrometers. It's a tenth of a millimeter, isn't it, with the naked eye, at a push. I probably couldn't at my age, but maybe with my glasses. Anyway, the researchers go on. They were animated worm-like entities chains, spirals, tubes, right-angled structures containing other artifactual entities within them. Artifacts within artifacts. All these are exceedingly beyond any expected and acceptable level of contamination of the Covid-19 injectables. Well, I would have thought 3 to 4 million per mil is way off the scale. Indeed. And incubation studies revealed the progressive self-assembly of many artifactual structures. What the heck are they? As time progressed during incubation, simple 1- and 2-dimensional structures over 2 or 3 weeks became more complex in shape and size, developing into stereoscopically visible entities in 3 dimensions. These became 3-dimensional structures in their incubated cultures designed to replicate the conditions inside the human body. They resembled carbon nanotube filaments, ribbons, tapes, some appearing as transparent thin flat membranes, others as 3-dimensional spiral beaded chains. Some of these seem to appear then disappear over time. Our observations suggest the presence of some kind of nanotechnology in the Covid-19 injectables. Now, this is directly from peer-reviewed literature. I don't expect you'll see it. I suspect strongly that I am currently talking to myself in a back room in Carlisle somewhere. If some of you do get to see it, then we consider that a bonus. You can think about it. Now, I'll just give you some of the references here for this. So this is the journal here, International Journal of Vaccine Theory, Practice and Research. The International Journal of Theory, Practice and Research is a peer-reviewed, scholarly, open-access journal concerning the development, distribution and monitoring of vaccines and their components. All content is freely available without charge to the user or his/her institution, which of course is excellent. Users may read, download, copy, distribute, print, search or link the full text of articles or use them for any other lawful purpose. Permission is not required from the publisher nor from the author, but we do give them full credit, of course. Isn't this refreshing? This is our work, we're reporting on it, and it's free, it's in the public domain. It is the antithesis of the control agenda that so often we've seen, with people not wanting to release papers for decades after the research was been done. Very refreshing. So that is about the journal there. The actual article itself is, have we got the article itself? What do I do? Oh, there we go. That's the journal there. That's the full journal. And again, you can download the full journal. Quite excellent. So that's the paper. That's the journal. I've downloaded it in PDF, of course. And that's the attribution, non-commercial, non-directive deed. I can't quite fully understand that. But it's great that it's basically share. You are free to share. And it's just, and of course we give full credit to the authors, which I will do now. These are the authors here. Physician Dr. Youngmai Lee, Republic of Korea, and Professor Browdy, Okinawa Christian University, Japan. And of course, the links, as always, are there to check them out for yourself. So what we've done is report on a peer-reviewed paper. It's in the scientific literature, as we've completely demonstrated in this video. I'm just sorry that I suspect that not many of you will get to watch this video, but there we go. Yeah, isn't that transparency refreshing, and really refreshing to see that? This is our work. Look at it, share it, agree with it, disagree with it, do further work to prove it, do further work to say it's a load of rubbish, but it's there, open for public peer analysis. Brilliant. The antithesis to so much of the more commercial research of the past few years and decades. But for now, look at the paper for yourself. Let me know what you think, if any of you actually get to see it. So bye. Hope I'm not talking to myself, but thank you for watching if somehow you have.